It is 3 o'clock. Let's go ahead and get started. I am Professor Heckman. You are not going to see my face today because I'm recovering from a terrible allergic reaction that I had to a medicine that I took for a previous illness that I had. And my face looks like Cthulhu and the Swamp Child had a love baby or something like that. So anyway, we're going to start with the PowerPoint, and then we're going to move on to the lab. And all of the things that that we're seeing today, you can find in modules. I'll go to student view. You will see I've already posted the test 1 study guide, so you can get started on that. This is what we're doing today, a chapter 1 experimental design. I have the PowerPoint here for you. And for today only, I'm giving you my version of the PowerPoint. Let me just go turn off this. So typically I have student versions of the PowerPoints, and you will download that to your computer before class. And then during class, you will follow along and you will fill in some blanks that are going to be on your version of the PowerPoint based on what you see on my version on the screen. But today I have given you my version. We're going to do some variables practice right here. There's one assessment question for the end of the chapter that you guys do. And then this is the lab that we'll be doing. So let's go ahead and dive right into the PowerPoint. I love to start the semester with this quote by Jules Verne, who said, "Science, my lad, is made up of mistakes, but they are mistakes which it is useful to make, because they lead little by little to the truth." And I think this is a beautiful way of looking at science, because we always think, when you do an experiment, there are results that you hope you get. And if you don't get those results, you often feel like maybe you didn't do something right in the experiment, or maybe something was wrong with the materials. And you learn more about analyzing a failed, quote unquote, failed experiment than you do about celebrating your successes, which is also important. But just keep in mind that as long as an experiment yields data, it's a good experiment, as long as you learn something from it. First of all, if you're sick, stay home. I do track attendance, but that's only because that is an NWAC policy. It is not part of your grade. But if you do not attend, obviously you will not get a good grade in the class. Attendance is essential to pass the course. You will not see me today. This is what I look like normally. This is my special interest. I love turtles and tortoises. This is a giant Galapagos tortoise at the Oklahoma City Zoo right here. And this is my family. That's my dad. He's Dr. Heckman. And there's my mom, who's Mrs. Heckman. Please don't call me Mrs. Heckman. It makes me feel old. There's my brother, Matthew, and Luke. And then we have my sisters, Rebecca, Biz, and Esther. And then that's me. I'm Professor Heckman. These are the most important men in my life. On the left is Wolverine, and on the right is Loki. They're little terrors, little hellions, but they're cute, so they get to stay. Throughout the semester, you will notice that I sweat a lot. I sweat at random times, and I sweat more than most people do while they're exercising. This is because I have a condition called hyperhidrosis, and I know that a lot of you are in this class because you want to be in the medical health field, whether a nurse or doctor or some kind of technician. So, and 3% of the US has hyperhidrosis, so I thought I would give you a little education about it, since if you do become a public health official, you're probably going to see somebody with hyperhidrosis at some point in your career. Basically, what it means is excessive sweating at any point triggered by random nerve signals. So typically, sweating is triggered by increased surface temperature or exercise. But if you have hyperhidrosis, it's just triggered by the fact that you exist. There's two different kinds of sweat glands. Oh, wait, I'm getting ahead of myself. Risk factors include genetics, repeated feverish infections, if you had it as a child maybe, history of tuberculosis, or chronic alcohol consumption. And there's a lot of celebrities that have spoken openly about their issues with hyperhidrosis. We have Charlize Theron, David, I think that's David Hasselhoff. We have Halle Berry, Steve Carell, Robert Pattinson, Chrissy Teigen. All of these people struggle with hyperhidrosis. Basically, there's two kinds, primary and secondary. Primary is caused by a malfunction of the nervous system. There's two kinds of sweat glands in skin tissue. We have eccrine sweat glands and apocrine sweat glands. The eccrine sweat glands are the ones that go directly to the surface of the skin, whereas apocrine sweat glands reach through a hair follicle. So nerve signals typically tell sweat glands when to produce sweat. But if the nerve signals are faulty, they're going to produce sweat at any time, predictable or not. Secondary hyperhidrosis is typically the side effect of some kind of medication that you may be taking. There is no cure for hyperhidrosis, but you can change your lifestyle to manage it. There are powerful antiperspirants about, or you could eliminate everything good in your life, which means spicy food, caffeine, and alcohol. So those are some interesting ways to manage your lifestyle. My cousin has this in her hands, and this is what she does. She uses iontophoresis, where she basically puts her hands in these little pans of electrolyte fluid and electrocutes them. And that stuns the sweat glands, the excessive sweat glands in her hands. And it doesn't work very well, but it works better than nothing, so she continues to do it. There are some other medical treatments that are available. You could get Botox injections. That is a temporary thing. Botox paralyzes the nerves that lead to the sweat glands, so it prevents them from receiving those nerve signals. But you have to get that redone every 6 months. You could do something more invasive, some surgery to remove the sweat glands. You could go up in there and cut the nerves that connect the sweat glands to the brain, or you could get them removed through laser surgery. You could also use microwave therapy to destroy the sweat glands using microwave energy. This is my bibliography. You guys are going to do a final project this semester, which will be a presentation about an endangered animal. And as part of that project, you're going to learn how to cite things in the APA format. And all of my citations are in APA format, so you'll get pretty used to it before you have to do it. Let's talk about how to succeed in this class. First of all, come to class and pay attention and take notes. You will need a laptop. If you do not have a laptop, there are some available for use in the classroom only. You can use those. You'll have to bring a flash drive or something to transfer your files from that laptop to your computer back home, or at least to your purse, because you don't know if you're going to get the exact same laptop the next time you come to the class, because they're all kept in this kind of big bin. But you're going to need one of those. Read over your notes after class, even if it's just a quick reread, and use the study guides to prepare for the test. It helps to start working on them a week before the exam at least, or at least even a few days, but definitely not just the night before. So the first study guide is already up, and after today, you'll be able to go in and fill out everything under the Chapter 1 heading. So you could already get started with that today if you want to. You don't have to, of course. Syllabus. The syllabus is long, but that's because it is my contract with you. It contains makeup procedures for exams and assignments, our participation, attendance policies, all kinds of things that you need to know about, and you are responsible for knowing the information that is in that syllabus. A recording policy, if you would like to record the meetings, now I'm thinking Microsoft Teams. If you would like to record the lectures, let me know and we will work something out. And let's go over some highlights of the syllabus, shall we? The very first thing, don't be afraid to ask for help when you need it. I became a teacher to watch my students succeed, not to watch them fail. This is my email address, aheckman, and my last name, Heckman, does have 2 N's on the end, so make sure you put a second N there, or it will go to somebody else. Technology computer, we already talked about that. Microsoft Word and PowerPoint. You will need Microsoft PowerPoint in order to edit the blanks in the PowerPoints that I give you. And this shows you how you can download it for free. Lab manual, we will talk about that in a minute. Most of you have been able to print the lab manual, but some of you have not, which is understandable due to, accessibility issues with the weather and everything. But I think that if you, oh gosh, where was I going with that? I need more coffee. Hold on. All right. Today I have made a typable version of the lab handout for Lab 1. So you will not need your lab manual today. And I have changed the due date on bringing it into class until next Monday, a week from today, which is the 29th. You will bring that in and you will get 15 points for having it. This is just to prevent panic printing at the beginning of a class period that overloads the library and sometimes it backs up their machines. And so then everybody needs to print something and the machines are backed up and it's just no fun for anyone. If you have COVID-19, don't come to class. Course goals, learning outcomes, exams and quizzes. We'll go over this in a minute, but basically they will all be taken in person on the scheduled date. Yes, they will be taken on computers, but because this is a face-to-face class, we are required to do the exams in a face-to-face format. So you're just going to be working on laptops. And we will use HonorLock in the classroom to prevent people from switching between browser windows and the quiz, which has happened in the past. We'll talk about that in a minute. Labs, we're going to do our lab safety protocols in a moment. There are no makeup labs because each lab requires setup and use of specific materials, which may be dangerous or perishable, and they are provided by the school only during lab sessions. So makeup labs are not possible. But I do understand that things come up and you might have to miss a day, so I dropped the lowest exam score, lab score. So if you get sick or if your child gets sick or something like that, you will have to take a 0 for that day, but it won't affect your grade because I dropped the lowest lab score. Here's our grading scale. I also dropped the lowest exam grade, but I do not drop the final. You must take the final. Assignment expectations. Write legibly so I can read something. If I can't read it, I can't grade it. If you miss a class, contact a classmate to find out what you missed and what assignments you need to complete. And this will be easier once you guys have each other's contact information. During our next class, I hope to be able to do our icebreaker, which we typically would already have done, but it helps you guys to get to know each other, and then you share contact information so you can form study groups, and a lot of people form group snaps and things like that. Sometimes we do things in class for a grade, and if that is the case, you have to be in class to get that grade. If it is an assignment which can be made up, you can make it up, but sometimes they can't be made up because they're group work. So just be sure that you come to class whenever you're able. Check your email. Sleeping. If you need to go to sleep, go sleep somewhere else. Do not sleep in the classroom for 2 reasons. First of all, it's rude. And second of all, it's ineffective. Sleeping works best when you're lying horizontally on a soft place and it's dark and it's quiet. And that does not describe a classroom environment at all. So go find somewhere soft to lay down for a few minutes and come back when you're feeling better. Come to class alert and ready to learn. No phone conversations during the class. If you have to have a phone conversation, that's fine. Just go outside in the hall. Attendance and participation. If you're sick and maybe contagious, stay home. Don't bring your germs to the classroom. Plagiarism. There's some information about plagiarism here, but it is not tolerated, basically. How to succeed. We've talked about that. College policies. Administrative drop policy. Okay, you must attend and fully participate in at least 2 classes. Okay, well, that is lax, but you have to at least participate in 2 classes, period, and complete the lab safety quiz before the 30th of January, or you may be dropped from the course for non-participation. That allows you to receive an 80% refund on your tuition if you're not participating. We have free mental health counseling for the students. There's the link for that. There's also a food pantry on the Bentonville campus. And then the most important part of this syllabus is the back page. We have the schedule. It is your responsibility to know these dates and be prepared for all the tests. And then down at the bottom, we are section 11. So y'all's final will be on May 7th from 3 to 5 p.m. And that is our syllabus. Let's talk about exams. If you do not take an exam, you will receive a grade of 0 because you did 0 work. Only 1 exam per semester may be missed and made up. If you want to make up an exam, you must contact me at least 40 hours before the exam to schedule an alternate time. If you have an emergency the day of exam, you must contact me by email before the start of class. Okay, so that means the timestamp of the email has to be before the start of the class. If you contact me anytime later than that, you will not be allowed to make up your exam. And you might have to, go and do something really important, but you are paying for your education. You are invested. You want to put some effort into this, so you can afford to spend 2 or 3 minutes to send an email about the fact that you're going to be late in order to allow yourself to make up that exam. Let's download the app Socrative Student. Go to Google Play or your Apple App Store. And this allows your phone to function as a clicker device. It's going to ask you for a room name. Our room name is NWAC Bio. And we're going to do some practice clicker questions. I really like to have these clicker questions because they keep people engaged and it gives you an opportunity to use your phone, and we all like to use our phones. Okay. See, we've got 8 people in here. All right. 10 people. Awesome. All right, let's go to the first question. All of these scenarios are things that have actually happened. A student approaches me the day after the test and says, my mom had to go to the hospital yesterday and I couldn't contact you. When can I make up the test? Will the student be able to make up the test? Yes or no? All right, excellent. Wow. 100% chose B, which is the correct answer. Yes. Is this working? Excellent. Yeah, he will not be able to make up the test, but there is something he could have done to allow himself to be able to make up the test. What is that thing? Okay, 92% chose the correct answer, which is B. Well done. And here is our final, no, wait, this is not our final clicker question. This is our next clicker question. What if a student does not take up the test, does not contact me? Will the student be able to make up the test? Yes or no? Yes, 100% chose B, which is the correct answer. Good job, guys. Here is the final clicker question. If a student emails me 48 hours before the test, says, I'm not going to be there on test day, when can I make up the test? Will they be allowed to make up the test? Yes or no? Interesting. Okay, they will be able to make up the test. Yes. 86% chose A, which is the correct answer in this situation. All right, so that's all with the clickers for now. We'll do some more of that later. Course formats. If you have COVID-19, don't come to class. Oftentimes, I will give you short quizzes before class, and I give you the answers to those quizzes the day before. I do this at the beginning of the class period, so you need to be at class on time, A, to get the answers for the next day's quiz, if there is one, and B, to hear any announcements that I might be making. I give you these quizzes because it helps to keep up with the material, helps you to review, gives you an idea of what might be on the test. And if you miss class, you can always make up a quiz, but you are responsible for finding out if there is a quiz, when their quizzes are, and what the answers are. And this will be easier again once you guys have each other's contact information. You are responsible for printing all class handouts. I cannot afford to print everything for everybody. All class handouts will be under that day's module in modules. I've already told you about the PowerPoints, which you will download. There are some blanks in them for you to fill out. If you are the kind of person who likes to read the textbook, I have provided some active reading guides for you. These are optional. I will take a look at them and grade them for you if you like, but they are not for a grade. And I don't assign reading from the textbook because I understand that not everybody can afford it. But it does have a lot of good information in it. And it's where I draw it's my source material, basically. So I do encourage you to read the textbook, but it is not necessary. Safety, lab safety PowerPoint. Yes, most of you have already done the lab safety quiz, except for Jocelyn and Grayson. You two need to still take the lab safety quiz before you begin the lab today. So I'll give you a little time to do that. And so most of you have gone over this lab safety PowerPoint, but guess what? It's college policy to do it again. I have to do it with you. So let's begin. Principles of Biology Safety Lecture. Ground rules: no food or drink allowed in the classroom, because the classroom is our lab area and we can't have food and drink in there. You don't know what people have been spilling or working with on your desk before you. On lab day, you must be in pants and closed-toed shoes. If you are not properly dressed for lab, you could be sent home. So we have scrubs and we have booties that you can put on over your clothes if you don't remember to wear appropriate clothes. But if we run out of those things, then you will have to be sent home to change. This is just a safety issue. If you are in doubt about a proper laboratory procedure or given task, you should ask me before proceeding. Lab stations should be kept free of extraneous items. So your desk will be your lab station. Typically, we work in groups of 4, and everybody will gather around the desk. And so you want to keep, it'll get pretty crowded, and you want to keep your extra stuff off of the desk and your phone especially. After your lab is finished, you should clean your lab station with Simple Green. We actually do not use Simple Green anymore since COVID began. We use a hospital-grade disinfectant, which kills coronaviruses. Lab equipment, make sure it's clean and put away at the end. Make sure you've rinsed all of your test tubes. Shoes with closed toes and backs are required. So don't wear shoes with cutouts or vents. Long pants, you cannot wear pants that have tears or holes in them. You will have to put on scrub bottoms over those. And you also don't want anything that is really tight, because when a fabric is spilled, like when water spills on a fabric or any kind of chemical, a liquid chemical of any kind, that fabric is going to soak it up. And then if it's tight fabric, it's going to hold it right next to your skin. And so you don't want to be wearing leggings that are holding like some kind of acid next to your skin instead of jeans, which would be holding it a distance away from it. Do not wear loose-fitted clothing because you might drag your sleeves through something and knock it over or get chemicals on them. Don't wear dangly jewelry. Basically, don't wear things you are not willing to part with, because if you get a hazardous chemical on your clothing, we will have to take it and have it sent off to be properly destroyed. Do not wear high heels or wedges, because those are presenting a greater tripping hazard than flats. Tie back your long hair so it doesn't get caught in stuff or catch fire. PPE stands for personal protective equipment, gloves, and safety glasses. It is very important to wear your PPE. Gloves are provided at no expense to you. They are free. There are as many of them as you could possibly need. So please, please use the gloves. They are to protect you because you will be working with things that are carcinogens, things that are hazardous in all kinds of ways. Sometimes they'll be volatile, which means they have a tendency to explode or catch fire. They could be poisonous. We use nitrile gloves. If you need a kind of glove other than nitrile, let me know. We have some other options, but nitrile gloves are completely latex-free. You have to keep in mind, though, when you are wearing the gloves are to protect you from other stuff, but it does not protect other stuff from you. So if you are wearing your gloves and you're messing with a chemical, and then your phone buzzes and you go over to touch your phone, whatever you were messing with is now also on your phone. So just be very cognizant of that. And if you need to take off your gloves, then that's fine. Just take them off and throw them away and put on another pair. Yes, if you're allergic to nitrile, let me know. Safety glasses. If you already wear glasses, you do not have to wear safety glasses. But it's if you don't wear glasses, you will have to wear them. Wafting. If you need to smell a chemical, don't stick your nose over it like this person in the first picture. That's the wrong way to smell a chemical. The right way to smell a chemical is to keep a distance between yourself and the chemical, and then use your hand to drift some of those vapors towards you, and in that way, you can smell the chemical. This man says, I don't always smell chemicals, but when I do, I waft them. Chemical reagents. A reagent is a chemical that you are using for an experiment. These should be transported with 2 hands. Always be aware of your surroundings so you don't trip and fall. Don't carry more than 1 chemical at a time. I don't need to see people loading up their arms with little bottles and vials and stuff and then wobbling over to the lab bench and plunking it all down and then little vials rolling off onto the floor and stuff, because we don't need any of that on the floor. If you spill something, let me know immediately so we can clean it up appropriately. SDS sheets, safety data sheets. These tell you the dangers that are associated with a chemical. We have them in 2 places. There is one binder at the side of the classroom that contains all of the SDS sheets for the entire semester. And then at each lab, there will also be a binder containing the SDS sheets for that lab specifically. If you break glass, do not pick it up with your hands. We have a little dustpan and a little broom. You're going to take those, sweep up all the glass shards you can find and put them in. There's this cardboard box that I'll show you. It's by the refrigerator in the classroom. And let me know if you break something, of course. If you've got blood on the glass, then that has to be put into a biohazard container, which we also have. If you get an injury, any kind of injury, even if it's just a little scratch, you should report that to me because it could become something big. And if it becomes something big, we want to have documentation of where exactly it started out. Accident reports. Yes, we will fill out the accident report. You'll sign it. If you are cut by broken glass, do not get yourself the first aid kit. Have someone else get you the first aid kit because you don't want to contaminate that first aid kit with your blood. And then you'll tell the professor, you'll tell me so that I can help you fill out an accident report. If you spill a chemical on your exposed skin, first flush the exposed skin under cold water for 2 minutes and let me know, and then we will reassess at 2 minutes. God forbid you get chemicals in your eyes, but we do have an eye wash station for that. It looks like this. You're going to find these little yellow loops and you'll flip them open to open the device. You'll push this lever towards the wall and that will start the flow of water through the eye spigots. And then you place your face over this little dish and you keep it there until we reassess after 20 minutes. If the chemical spill covers enough of your body that you cannot rinse the affected area in the sink or eye wash station, then you use the safety shower. This is the safety shower. You are going to strip. Everybody else leaves the room. You strip, and a friend will use the fire blanket that we have and hold it up as sort of an impromptu shower curtain. And you pull this triangle down, and that starts the flow of water. You stand under there for 15 to 20 minutes, and it is not warm. It's cold, okay? So try not to spill chemicals all over your body such that you have to use it. But we will reassess in 15 to 20 minutes. If there's a medical emergency, dial 911, obviously. If you are pregnant or immunocompromised, let me know, please. There are some labs that you will have to make accommodations for that. Allergies, if you are allergic to anything at all, let me know, because allergies, we often use food items in our labs. Chemical hygiene, label your beakers and test tubes and do not pick up unlabeled things. The fume hood. This is the fume hood. It's at the back of the classroom. And the fume hood is a device that has an internal circulation system of air. So whatever you put on in the fume hood, if it starts to vaporize, that vapor is going to stay within the hood. So if there's a chemical in there, it's in there for a reason. If you need some, do not take the chemical back to your desk. Instead, you're going to take your test tubes over to the hood, put some of that chemical in your test tubes, and then you'll take your test tubes back. But if there's something in the hood, it should stay there. Fire extinguisher. Use it on a small open fire, which is about 3 feet by 3 feet in size. Do not use it on a person. If people catch on fire, which I don't know why they would, but if they do, we have a fire blanket for that. Throw the fire blanket on the person, stop, drop, and roll. There it is. You would just pull that tab and the big blanket comes out. Fire extinguisher, this is how you use it. You pull the pin, aim at the base of the fire, squeeze the trigger, and sweep back and forth. If the fire is bigger than 3 by 3 feet, leave the building, go out of the classroom. And there's a couple of dumpsters next to the building or nearby the building that we will gather at. I will have to count everybody, make sure everyone's here. And do not crowd the parking lot because the emergency vehicles need to get in. If their fire alarms do go off, we go out, I will count you, and we cannot go back in until a police officer says that it's safe. There we go. Yeah, you must stay outside until a police officer or school official tells you can return. Now, these maps are for the campus in Bentonville. Our severe weather shelters are where the vending machines are and where the bathrooms are. So that gives you something to do. If a tornado comes, you could go to the bathroom or you could go get yourself a snack. It's up to you. More severe weather shelters, blah, blah, blah. If there is an active shooter, run, hide, and fight in that order. If you can get out safely, do so, obviously. Run and don't stop. Hide. If you're unable to safely exit the building, remain quiet. Lock your door, barricade the entrance, turn off all lights, and call 911. And fight as a last resort. Improvise weapons with the intent of distracting and disrupting the shooter. There is an app called BC Alert where you can get all major emergency alerts, weather alerts, school closings, etc. To sign up, you go to bcalert.com and click on sign up now, and then you will be able to subscribe to the NWAC feed there. The last things you should do before leaving the classroom are wipe your lab station down with disinfectant and wash your hands. Are there any questions? Okay. So the lab safety quiz not due tomorrow at midnight anymore. Nope. Lab safety, excuse me, the lab manual. This is the text that you have already seen of that announcement I sent out. I have changed the due date to the 29th. I think I already discussed this. Bring your lab manual on the 29th for 15 points. We're going to be skipping 2 of the labs that are in the manual, which is why I told you to print only these pages instead of the whole thing. And you must show it to me at the beginning of class. If you forget it and have to run off to print it, that does not mean you get credit, but you will still have to print it. So you might as well have it printed beforehand. Yeah, so take a picture of this slide, please. And that will help you remember. Okay. This is a Monday, Wednesday class. On Monday, the 8th, there is a once-in-a-lifetime event called a solar eclipse. A total solar eclipse is happening. I guess twice in a lifetime for us. And Arkansas is estimated to draw like many thousands of people. Like the population of the state is going to increase by like 150% because people from all over the country are going to come here. So that day we are doing a test. The test will not take the entire 3 hours. As I said before, all tests will be in person in the classroom, except just this one you will take on your own, and you will still use HonorLock. Breaks, let's get this out of the way. 10 minutes per hour of class time I'm required to give you for breaks. So let's go back to Socrative. Remember our room name. Let me edit this. Is MLAT Bio. Which break schedule would you prefer? Okay. 62% say A and 38% say B. So we are going with break schedule A, which means that I'm going to set some timers on my alarm here. On Monday and Wednesday, break is going to be 3:50 p.m., not a.m. And then there will be another break at 4:50 p.m. And then class will be over at 5:50 p.m. Okay. Yes, very good. All right, and it looks like, ooh, we have 10 minutes to break. Okay, perfect. So this is the first of those little quizzes that I'll be giving you at the beginning of class. This one will take place on Wednesday morning. Well, morning, quote unquote morning. The beginning of the class on Wednesday. I want you to get out your phone and take a picture of these things. Yes, spelling counts in college. You're adults now, you can do it. This is a beaker. It is used for holding, mixing, and measuring. A flask is like a beaker, except it has a narrower opening. It has a neck. A flask has a neck. This is a graduated cylinder. It has all these little graduations here, these little increments for measuring very precisely volumes of fluid. So take a picture of this slide. And then what we're going to do next time is I'm going to show you these pictures and ask you to write down what each thing is. Okay, and that is the little plastic part up there that keeps it from rolling. We also have test tubes, which are for small volumes of liquid, a test tube rack, and these little droppers, those are called pipettes. And those are used to move liquid from one place to another. So also take a picture of this slide. Maybe make those pictures the background of your phone or something, and look at them a lot between now and Wednesday, and you will do just fine on the quiz. And that will be 5 free points for you. Biology, the scientific study of life. Biology is the study of life, but what does it mean to be alive? Every living thing shares the following 7 characteristics. These are the 7 characteristics of life. 1, it has order on the cellular level, meaning it's made of 1 or more cells. It's orderly. It can reproduce. Oh, whoa, come back. So reproduction is important for the species as a whole, but not necessarily for an individual. So for example, if you have a dog and you get your dog fixed, your dog can no longer reproduce, but is still alive. Some individuals are born without the ability to reproduce because of infertility reasons. That doesn't mean that they're not alive. It just means they don't have this characteristic of life. So that one's important to the species as a whole, but not necessarily to each individual. And when I was teaching 8th grade, I gave this example, and a little boy in the front row folded his arms very seriously. He says, "Miss Heckman, when you get your dog fixed, the dog dies inside." I thought, oh, that's a lot of self-knowledge for an 8th grader. Okay. Anyway, all living things must be able to grow and develop. They must be able to process energy. That's called having metabolism. They must be able to maintain a stable internal environment, a set body temperature, a set pH, a set blood glucose level. They have to be able to respond to things that happen to them, and they must be able to evolve or change through time. The cell is the structural and functional unit of life. What that means is that every living thing is made of 1 or more cells. So let's think about some things that are not living. A drill, a handheld battery drill. That's not alive. Why not? Well, it's pretty orderly. Can it reproduce? No. Can it grow and develop? No. Can it process energy? Yes, it can do that through its battery. Can it maintain a stable internal temperature? Nope. Does it respond to stimuli? Yes, it does. When you pull the trigger, the little drill part goes around, but it cannot evolve or change through time. A living thing has all of these characteristics. How much time have we got before break? 6 minutes. Perfect. Let's talk about the process of science. Science is a method for studying the natural world using observations and testing. We cannot conclusively prove anything. And the reason for this is the same reason we cannot prove or disprove the existence of Bigfoot. So this is Bigfoot here, and we've got a picture of him, and that means that he must exist, right? Because we have evidence. But one piece of evidence doesn't make a conclusion. It's possible that this photo was doctored. It's possible that this is not actually a photo of Bigfoot. It's just somebody in a black ape costume. We don't know. We need more pieces of evidence. And in order to conclusively prove that Bigfoot does not exist, you have to search the entire known universe at the same time. Because if you just search one area and find that Bigfoot is not there, and then you move to search another area, well, Bigfoot could just have moved into the area where you had just searched and not found him. So you still don't think he's there, but he's there now. So you have to look at everything all at once, which is impossible. And that is even considering the fact that Bigfoot doesn't have the ability to become invisible, so that he does exist and we don't see him at any point in time, which admittedly sounds ridiculous, but we are talking about Bigfoot here, so I think we can suspend reality for a moment. So basically, there's always going to be an element of doubt, even if it's a very small element of doubt. And you can't conclusively, absolutely prove anything. Similarly, science cannot be used to test and explain everything, only the things that can be measured and observed. So here are two statements. The Killers are the best band ever, which is true, or the music of the Killers is listened to by many people. Which one of these is a scientific statement? The scientific statement is going to be the second one, because the first one is an opinion. It is different in everybody's mind, but everybody can agree that many people listen to the music of the Killers. That's a number. That's something we can measure. We can observe it. That's a fact. The existence of God is compatible with a belief in science. Not belief in science, I'm sorry. I'm compatible with practicing science. Just because God cannot be measured, observed, or tested does not mean God does not exist. It just means that science cannot be used to examine the existence of God. So here's our first clicker question of the lecture. Everybody go over to Socrative, please. Remember, our room name is NWAC Bio. Interesting. 47% say A. Oh, 43%. 24% say B. 32% say C. 4% say D. Oh, wow, now the numbers are changing. Oh, I guess maybe my phone was delayed in receiving some of the responses. Okay, 35% say A, 19% say B, 42% say C, and 4% say D. The correct answer here is actually C. And let's talk about why. Let's see, 32% of you said A, this peanut butter has unhealthy ingredients. Well, the word unhealthy can be measured because we know what is healthy and what is unhealthy. Unhealthy things include trans fatty acids, for example. We can look at the peanut butter and see if it has trans fatty acids. And if it has it or doesn't have it, that's a fact. You know, that's not going to be opinion. So we can test that using science. My dog is heavier than average. Well, we can find out what an average dog weighs of your dog's breed and age, and that will be a number that we can use to test that against. My mother-in-law is crazy. This is the science. This is the statement that cannot be investigating using science because there is no one definition to the word crazy. What I think is crazy may be different from what you think is crazy. I might think your mother-in-law is just a sweet, doting old grandma, and you might think that she's absolutely insane. But there is a way that we could change this statement to say, to be investigatable using science. Does anybody have any ideas? Okay, well, what we could do is we could just define the word crazy. And we could substitute that with a defined mental illness, such as bipolar disorder. So instead of saying my mother-in-law is crazy, we would say my mother-in-law has bipolar disorder because doctors have lists of criteria that people have to meet in order to be qualified for these diagnoses. So that could be investigated using science. And it is now break time, so I will see you guys. We will reconvene at 4 o'clock and continue. All right, everyone, it is 4 o'clock. Let's go ahead and get started again. Science, within science, we are always changing the way we understand and gather data, especially as new technology is developed, as we have better microscopes and better PCR machines and all kinds of newer, better equipment. We're able to analyze things on a more smaller and what's the word I'm looking for? Um, I don't know, tiny, I guess, on a tinier scale. And so the way that we understand things is going to change. And a great example of this that doesn't even involve technology is spontaneous generation, which is something that people in the 17th century believed life arises from non-life. Now we know that living things only come from living things. But in the 17th century, this is an actual recipe for mice here. It says if a soiled shirt is placed in the opening of a vessel containing grains of wheat, the reaction of the leaven in the shirt with fumes from the wheat will, after approximately 21 days, transform the wheat into mice. And we know that this is absurd. But if you did not know that mice had to come from other mice, and you did this and you found these results, it's a logical conclusion to think maybe the wheat turned into the mice. It's not true, but it is logical. Fortunately, like we have disproven spontaneous generation. We are more aware now of how things actually happen. Another example would be that in the 1800s, people thought protein was the hereditary material, which is the material that transmits traits from one generation to the next. But an experiment in 1952 found that DNA is the hereditary material. These are just some pictures of the experiment they did. And now that we know that DNA is the hereditary material, we can build on that. By manipulating DNA, we can manipulate our inherited traits. But not everything that any scientist spews out is going to be accepted as knowledge. It must be peer-reviewed and verified before being accepted as valid. Peer review means review and replication by fellow scientists, and verification is conclusion that results are valid. This is a great example of why this is important. This molecule is called polywater. It was discovered in the 1960s by one scientist, and it made a huge splash because it was supposedly a form of water that was denser than normal water, and it could change any water it touched into polywater, and it could not support life. So the concern was that maybe somehow some of this existed somewhere and it would get into the main water supply and would change all of the water to polywater, and then life wouldn't be able to exist because we wouldn't have any normal water anymore. Well, fortunately, no other scientists were able to replicate his results. Finally, one of them did, and he found that he was not analyzing water. He was not analyzing polywater. He was analyzing sweat water. The guy who did this experiment originally must have been sweating into his test tubes, because what polywater is just dirty water. It's of course it can't sustain life. You can't, live by drinking sweat water. That's gross. So this is why peer review and verification are very, very important. We can also build on previous knowledge. Now that we know that DNA is the hereditary material, we know about genes. We can manipulate genes, and through that we have genetic engineering. These are building on each other like steps on a step ladder. Knowledge progresses in order of certainty, from hypothesis to theory to law. Hypothesis is the least certain and law is the most certain. Hypothesis is just an educated guess. It does not have to be an if-then statement. We will not use if-then statements in this class. We will use null and alternative hypotheses. But basically, a hypothesis is any statement or prediction that can be tested. When a lot of hypotheses get together and analyze many different aspects of one specific theory, we all support that together. That is a theory. It's a well-tested explanation. It has to be supported by empirical data. Empirical data means data collected instead of just like guessed at. And it must be peer-reviewed and verified. before being accepted as a theory. So we have plenty of theories right now. The most one we're going to talk about the most is the theory of evolution. And the fact that evolution exists is not the theory. The theory is how it happens. So we're constantly learning more about how organisms change and evolve as they interact with one another. And so our concept of evolution, mechanisms of evolution, is changing. But evolution as a phenomenon is a fact. That's not necessarily a theory. And theories can be modified or expanded. The thing is that when you take knowledge to the level of law, that is not, you can't touch that. Like if you say this is a law, that means, okay, this is perfect. This is as good as it's going to get. We know this for a fact. So we have things like the law of gravity. But these days we don't make laws because we understand that our understanding of things are going to change as we investigate more. So we just make theories these days. Investigations, they have one of two purposes. They could be pure science, which is science for the sake of science, or applied science, which is applying knowledge towards a practical goal. Most grants and funds support applied science projects because those are the ones that are like working towards helping humanity solve different problems and things like that. But the issue is that applied science cannot exist without pure science. A great example is a spy probe that landed on, I think it was on the coast of California somewhere, and it had some grains of sand inside the probe. And scientists were able to go back to a pure science experiment that had been done, analyzing the composition of sand grains on different beaches in the world. Why had they done this? Well, like, why? Who would do that? Who would spend their time analyzing, the composition of sand on different beaches? There's no point to that immediately. There's no immediate point. But because of that knowledge existed, because of that knowledge, the scientists were able to apply it and figure out where this probe came from. Scientific method is a big circle. It's like a pizza. You can cut it into a different number of slices, but it's still a pizza. A different number of steps, still the whole process. I like to divide it into 7 steps. We're not going to spend a lot of time on this since everybody knows the scientific method. You make an observation, ask a question, form a hypothesis, do an experiment, analyze your data, draw a conclusion, and then finally communicate your results. This is the most important part of the scientific method, because if you don't tell other scientists what you found, then they can't benefit from your knowledge, and that knowledge can't be used to revise understandings and expand theories. So it's basically just one process that can be divided into any different number of steps. Now, speaking of hypotheses, we're going to have 2 types of hypotheses, null and alternative. The experiments always start with, does X have an effect on Y? The null hypothesis states that there is no effect. X does not have an effect on Y. The alternative hypothesis states that there is an effect. So null you can think of as nothing, no effect, whereas alternative is the opposite. Alternative is the opposite. So in this case, the opposite of null is something. It will have an effect. Here's an example. Will students score higher on a test if they study 5 hours instead of 1 hour? The null hypothesis would say that study time does not affect test score. And the alternative hypothesis is the opposite. It says that study time does affect test score. Then we're asked, what results would cause me to reject the null hypothesis? Well, if you are rejecting the null hypothesis, then that means you're seeing the opposite of the null hypothesis. So if we do this experiment and we see the opposite of the null hypothesis, if we see that study time does affect the test score, then we're going to reject that null hypothesis. And the same for the alternative hypothesis. If we see the opposite, we reject it. So if we do this experiment and we see that study time does not affect test score, we reject the alternative. Here's another example. Does the amount of food my dog eats affect his weight? Does X have an effect on Y? Null hypothesis says X does not affect Y. Alternative, X does affect Y. So a good null hypothesis would be the amount of food my dog eats does not affect his weight. Notice we are not talking about weight gain or weight loss, just whether weight is affected. It's really important when you are constructing your hypotheses that you have to keep them specific to the way the experiment was described. If I were to give you this scenario and ask you for a null hypothesis and you said, the amount of food my dog eats does not cause him to gain weight, that is an incorrect answer because that's not what the example wants to know. Affecting weight could mean gaining or losing. Alternative hypothesis would be that the amount of food my dog eats does affect his weight. And then what results would cause me to reject the null hypothesis? Well, remember, if we are rejecting the null, we are seeing the opposite of it. So the null says the amount of food my dog eats does not affect his weight. If we do this experiment and we see that the amount of food my dog eats does affect his weight, we reject the null hypothesis. If we do this experiment, we see the opposite of the alternative. So the alternative currently states the amount of food my dog eats does affect his weight. If we do this and we see that the amount of food my dog eats does not affect his weight, we reject the alternative. All right, this is Scott Pilgrim versus the World is a really great movie. Everybody should see it if they haven't seen it already. Scott Pilgrim is this little baby Michael Cera and his cute little blue-headed girlfriend Ramona are sitting in their basement eating garlic bread, and Scott says, I love garlic bread, you know, with his face stuffed full of garlic bread, crumbs hanging out everywhere. I can eat every day for the rest of my life and be happy. And Ramona said, will you be fat? He said, why would I be fat? And she said, well, bread makes you fat. And she says, bread makes you fat? Crumbs spilling everywhere. So let's go over to Socrative. And I'm going to start a short answer question. Remember, our room name is EnLAC-Bio. And type in what you think would be a null hypothesis for this experiment. Good, we've got some good answers coming in here. Yes, you must phrase it as a sentence. It cannot be a question. Okay, we've got some good answers here. Eating bread will not make you gain weight. Eating bread does not cause weight gain. Eating more bread does not have an effect on weight. Very good. Eating more bread does make you gain weight. That would be the alternative hypothesis. One person says bread does not make you fat. That is not phrased as a null hypothesis. A null hypothesis, you would say eating bread or eating more bread does not make you fat. Okay, good. Very good. So we've determined the null hypothesis is eating bread does not cause a weight gain. That's the null hypothesis right there. So if that's our null hypothesis, then what would you have to see in order to reject the null hypothesis? And I'll leave what in order to reject the null hypothesis. And I'll put the null hypothesis up here again. Okay. So now you've got our null up there. What will you have to see in order to reject this? Yes, you see the opposite. If eating more bread leads to weight gain. Very good. Yes, you guys got it. So the conditions to reject the null hypothesis are if you see that eating more bread does, oh, wait, it's already bolded. Oops, cause weight gain. So that would be the correct answer to this one. Eating more bread does cause weight gain. So now let's skip over to the alternative. What would be a good alternative hypothesis? Or nope, 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 nope. Yes. So remember, our room name is NWAC file. Very good. Okay. Yes. So the correct answer here is that eating more bread does cause weight gain. One person said eating more bread makes you unhealthy. There is not necessarily a direct correlation between weight and health. We have all kinds of things to consider, like body mass composition and if your fat to muscle ratio and things like that. And anyway, the experiment doesn't ask about healthy, it's asking specifically about weight gain. So you have to be careful to look only at what the experiment is asking for. But yeah, very good. So the alternative hypothesis states that eating more bread causes weight gain. Now, what about rejecting that? Okay. So if that's our alternative, then what are the conditions to reject the alternative? Let's restart here. Very good. Very good. Opposite results, yes. Eating bread gives you more fat food. That is not the correct answer because we're not talking about whether the food is fat or not. If eating more bread does not cause weight gain, no correlation between consumption of bread and gaining weight. Eating more bread does not cause weight gain. Very good, guys. You know your stuff. Okay. 421. Designing a controlled experiment. This is the last section of our lecture today, and then we will move into the lab. There are 3 types of experiments. The first is an observational experiment where nobody changes anything. And these are really useful for scenarios where you can only observe and you can't do a lot of manipulation. Like if you're working with endangered species, you can't kill one and then set it there and be like, okay, now what's everybody else going to do and watch what the other animals do. No, you just set an event in motion and observe without interfering. There's also a simple comparison where results are compared to one another, and that is in opposition to a controlled experiment where results are compared to one specific standard. We're going to talk about controlled experiments today. There are 4 parts to a controlled experiment: the independent variable, the dependent variable, the control or control group, and the confounding variables. Sometimes we call these variables held constant or constants. The independent variable is what I change. It's manipulated, and it is the cause of the change. The dependent variable, this is the effect. This is what we're measuring to see if anything happened. You could insert them into one of these sentences to help you find them. Does independent variable cause a change in dependent variable? Or you could use this much longer sentence, but I don't like that one as much. So let's do an example. Trees receiving the most water produce the largest lemons. What is the independent variable and dependent variable? What is the cause of the change in this sentence? Trees receiving the most water produce the largest lemons. That's going to be the amount of water, not just the water. We can't just say water. That's not specific enough. Well, there's anything about water that could, like the composition of the water, the source of the water. Maybe if it's bottled water, the brand, which I don't know why you would water a tree with bottled water, but you know, whatever, people do things. So the amount of water is the independent variable. That's the cause. And then what is the effect? The size of the lemons. That's the effect. So that's going to be the dependent variable. Oh, here we go. You also have a control. This is a group without the independent variable. It is used as your standard. This is what you're going to compare all the rest of your results with. And then you have your confounding variables or your constants. These are things you're not testing that could affect the outcome if they were changed. You don't want them to get in the way of your results. So let's do an example. More pounds of potatoes will be produced if the soil is fertilized more. And this is similar to what I will give you guys on an exam. I will give you a statement like this and ask you to identify the parts of the experiment. So let's take a look. More pounds of potatoes will be produced if the soil is fertilized more. First, we have to analyze the variables, independent and dependent. So these are the things that are changing. These are the variables, the pounds of potatoes and the amount of fertilizer in the soil. Let's say the cause is going to be the amount of fertilizer in the soil. Yes. You can't just say soil, though. What about the soil? The source of the soil, the composition of the soil, the amount of soil. No, we're studying the amount of fertilizer in the soil. Similarly, you cannot just say fertilizer. That is an incorrect answer. What about the fertilizer? You have to say the amount of fertilizer because there's many different kinds of fertilizers with different compositions and different nutrients. The dependent variable is going to be the effect, so that's the pounds of potatoes. You cannot say potatoes and you cannot say pounds. Both of those are incorrect answers because they are incomplete. Pounds of potatoes. You could also use our sentence, does independent variable cause a change in dependent variable? And what we do is we fill in the blanks with the variables until the sentence makes sense. So let's try it this way. Does pounds of potatoes cause a change in if the soil is fertilized more? That makes no sense. Let's try again. Does if the soil is fertilized more cause a change in pounds of potatoes? There we go. That makes sense. So now we know that the pounds of potatoes is the dependent variable and the amount of fertilizer in the soil is the independent variable. The control is a group without the independent variable. So the control group would be potatoes grown without fertilizer in the soil. So those guys are going to be your standard, and you're going to compare all of the other potatoes to those potatoes. Confounding variables. All of the potatoes are going to need to get the same amount of sunlight. You should use the same species of potato. You should give them the same amount of water. There's many more different examples. Those are the ones that I came up with. Null hypothesis states that independent variable does not affect dependent variable. So the amount of fertilizer in the soil will not change the pounds of potatoes. And the alternative states that independent variable does have an effect on dependent variable. So amount of fertilizer will change the pounds of potatoes. So these are those correct answers for that one. Let's do another one. I'm going to set the timer for 5 minutes and have you guys do this. And where's my 5-minute timer? And we will talk about it in 5 minutes. You can just write it on a piece of paper. You're not going to turn this in. All right, let's go over this. The growth rate of a pine tree depends on how much carbon dioxide it receives. First, we figure out the variables, the cause and effect. The cause here is how much carbon dioxide the tree receives. So that's going to be our independent variable. The amount of carbon dioxide. Again, if you just say carbon dioxide, that is not a correct answer because it's not specific enough. We're talking about the amount of carbon dioxide. The effect is the growth rate of the tree. Again, you can't just say growth. You have to say growth rate of tree. The control. There's two possibilities here. If you were actually doing this experiment, which people do, they do this experiment up in northern areas where there's a lot of pine trees, they just take massive amounts of carbon dioxide and dump them on top of these plots of pine trees. And their controls in that situation are trees with unaltered atmospheric normal carbon dioxide levels. For the purposes of this exercise, I would also accept a tree without carbon dioxide as a control, because the control typically is a group without the independent variable. In real life, that would not work, but we're just doing an exercise here. Confounding variables, you will need to have the same species of tree, the amount of sunlight that the tree gets, they will all have to get the same amount of sunlight, they will all have to get the same amount of water, so on and so forth. There's many different confounding variables. The null hypothesis states that the independent variable does not change the dependent variable. The amount of carbon dioxide a pine tree receives will not change its growth rate. And the alternative states that the amount of carbon dioxide a pine tree receives will change its growth rate. It's the exact opposite of the null. All right, so that brings us to the end of the lecture portion. I always like to try and have some interactive worksheets, and obviously we unfortunately cannot do the interactive portion here, but we can still do the worksheets. Go over to the modules in Canvas and click on Variables and Experimental Design Practice, which will be in Chapter 1. Our Chapter 1 module, we're going to download that. Oh, it looks like I've already got it. Well, I'll download it again. Okay. You are not going to turn this in. This is just for practice. Go ahead and answer these questions. I'm going to set the timer. This typically takes about 20 minutes. So I'm going to set the timer for 20 minutes. Get as far as you can. And we will reconvene in at 4:36, 4:56. And we will talk about these answers. And I'm sitting right here if you have any questions. All right, it is break time, so you can either continue working till 5 or you can just stop where you are, go take your break. But either way, we will begin again at 5 o'clock. All right, it's 5 o'clock. We're going to get started again. Let's talk about the answers to this. Variables and experimental design practice worksheet. Numbers 1 through 3, we are identifying the independent and dependent variables. So the independent variable is the cause. No, what was that? And what is causing a change here? Apple trees grown in full sunlight will produce the largest apples. So the amount of sunlight is what's causing the change. You cannot just say sunlight. Sunlight is an incorrect answer. All right, and dependent variable, that's the effect. So what's the effect here? The effect is not the apples. Apples would be in effect regardless of the amount of sunlight. We're talking about the size of the apples. You can also not just say largest apples because you're not only measuring the largest apples, you're measuring all of them. Okay, number 2, an investigation was done to see if corn seeds would sprout at different times depending on the temperature of the water in which they were placed. Independent variable first. That is the cause. That would be the temperature of water. You cannot just say temperature and you cannot just say water. That's not specific enough. Dependent variable would be the time to sprout. Again, if you have questions, like if we were in the classroom, I would be able to look at you guys and gauge confusion, but I can't do that. So you're gonna have to speak up if you have a question. Number 3, the amount of pollution produced by cars was measured for cars using gasoline containing different amounts of lead. What is the cause here? The amount of lead in the gas. You cannot just say lead. What about the lead? The amount of lead in the gas. And the effect that we're having, the dependent variable, is the amount of pollution produced. Okay. Number 4, gosh, I feel bad for these plants. 3 sunflower plants are watered with salt water. Each plant receives a different concentration of salt solutions. A 4th plant receives pure water. After a 2-week period, the height is measured. What is the independent variable? So the independent variable is the cause, is the amount of salt in the water. We could also say concentration of salt solution. That would work as well. Dependent variable is going to be height of the plant. Possible control. All right, so the control is what you're comparing all your results to. In this experiment, they tell you what the control is. A 4th plant receives pure water. So that's the plant without the independent variable, basically. All right, plant with pure water. And a possible constant. That could be anything. So all of the plants must receive the same amount of sunlight. They must receive the same volume of liquid, whatever liquid they're happening to get. They must be the same species of flower. There's many different answers for you to go for that one. Number 5, 1 tank of goldfish is fed the recommended amount of food per day, according to the instructions on the fish food package. A 2nd tank gets twice the recommended amount of food, and a 3rd tank gets 4 times the recommended amount. For 6 weeks, the weight of the fish is recorded daily. The independent variable, the cause here, is the amount of the food. The effect is the weight of the fish. Control, they describe that here, is the tank of goldfish fed the recommended amount of food. For the purposes of the exercise, I would also accept goldfish fed no food because that is technically the group without independent variable. To actually do this experiment, you're going to have to feed your base goldfish some amount of food. But for the sake of the experiment, for the sake of the exercise, I would accept that answer. Constants, some things, they all have to be in the same kind of tank. It has to be the same food that you're giving them. Let's see what else. Same water quality. Possible null hypothesis. Null hypothesis says that independent variable does not affect dependent variable. So that would be amount of food does not affect the weight of the fish. Alternative hypothesis states that independent variable does affect dependent variable, which would be the amount of food does affect the weight of the fish. To reject the null hypothesis, if we see the results that are the opposite, if we see the opposite. So if we see that, what's the opposite of the null? The null says amount of food does not affect the weight of the fish. So if we see that the amount of food does affect the weight of the fish, then that is the conditions that we would reject the null. And accepting the null is if we see what the null predicts. So if we see the amount of food does not affect the weight of the fish. All right. Are there questions about this? Okay. I'm going to close that up and close this up. And now, oh wait, I closed it too early. No. Okay. There is one last thing. On this very last slide here, you guys have this again in your PowerPoint that I have given you in the modules. This is the final assessment question. It's just for me to assess how well you have understood what I taught you here today. And in order to do this, we're going to go over to modules, just like this. Again, we're going down to our Chapter 1 module. There it is, Chapter 1 assessment question. And it's going to ask you to identify each part of the experiment. There's no time limit. It's open response, but it's going to be due, oh, I need to change that, due at midnight. So let's make that due at 11:59 p.m. There we go. All right, so you need to do that sometime between now and midnight, and it will close at midnight. What that means is that if you have started it and you are in the middle of it at midnight, it will close you out. So be sure you start with enough time to finish. And next, we're going to go back to modules. We're going to start the lab. This is an online version of the lab we would actually do if we were in class. Going down to our, oh, I should go back to student view. There we go. Down to our module here. All right, so you're going to click on this link, Lab 1, Scientific Methodology Online/Remote. Here are the instructions, okay? This is the PowerPoint presentation that I'm going to give you right now. And then, where's the link? Oh, I didn't put the link. Okay, so let's just do that. Okay. Here is a typeable document for Lab 1. And I'm going to make sure we're going to put document there. Is all right, so that should be there now. There you go. So now when you click on that, you'll see a document that you can type your answers in. You will have to hand draw a graph and upload a picture of it. But I mean, this is as good as it's going to get. If you have it printed already, you can use the graph paper that's on there. And then these are the instructions. This is the PowerPoint video or presentation that I'm going to give to you right now. Scientific methodology. In today's lab, you can skip page 4. Page 4 is not for us today. First, you're going to do an observational experiment, or at least you would have done. That's what the videos are. You're going to watch somebody do the experiment. And then you would design an experiment to test your own hypothesis. Remember, there's 4 parts to a controlled experiment. We have the independent variable, which is the cause, the dependent variable, which is the measured effect, the control, which is the group without the independent variable, and the constants are things you're not testing that might affect the experiment if they were changed. Our hypotheses are the null, which predicts that independent variable does not affect dependent variable, and alternative, which predicts that independent variable does affect dependent variable. This is what you would see on the lab cart. These are the equipment pieces that we would be using: our graduated cylinder to precisely measure liquid, our thermometer to measure temperature, and our beaker to hold the liquid in. So what you would do is you would gather all your equipment here. You would measure the amount of liquid out into the graduated cylinder and then pour it into the beaker and measure the temperature. And then you would record the temperature. And you would put the tablet in, the Alka-Seltzer tablet, and start timing. Now, you do not want to wait until all the bubbles disappear. You will be waiting for hours. You call time when the top of the water clears, okay? Not necessarily when all of the bubbles disappear. On page 7, for the graph, this is some sample data. This is not your data. You are not going to graph your data. You're going to graph this sample data that another class did. You're going to calculate their average time, and then you're going to put it on a graph. This is what I will be grading when I grade your graph, because all graphs have 4 things. They first have a title. The title goes on top. It shows you what the graph is showing. The purpose of the title is so that you can understand what the graph is showing without having to look at the rest of the text. So a title for this graph might be time required for Alka-Seltzer to dissolve in different temperatures of water. That is a long title, but it is complete. It is descriptive, and it shows what's going on in the graph. Next, we have labels on the axes. We have the independent variable on the x-axis and the dependent variable on the y-axis. Then we have our units in parentheses next to the labels, just like that. And finally, we have our data. If you are using a line graph, you always connect the dots. You will be graded on that. There you go. So there's our data. On page 7, there's a typo. It says, why is it important to collect more than one set of data for each temperature? Why compute an average temperature? What it means is why compute an average time? Because we're doing that over here, computing an average time. We're not computing an average temperature. And finally, you're going to upload a scan or pictures of pages 5, 6, and 7 to Canvas. If you have used the typable document, you can just attach that and then upload a picture of your graph that you drew. This is what we would do to clean up in the classroom, but we're not in the classroom, so we're not going to do that cleanup list. And now you can go and start that lab. It's due by midnight. So by midnight, you have 2 things. You have the assessment question and you have the lab that are both due. And I will be here until 5:50 because that's when class is over, to answer questions if you have them. If you are done, you can leave now. If you want to stay and work on the lab and ask me questions, that is something you can do as well. And thanks for coming. Hopefully I'll see you guys Wednesday.